Hey sqadians, do you know what is Major Histocompatibility Complex? If not, then no worries. Like always, we are here with another amazing short scanimation video to introduce you with such an interesting topic that is Major Histocompatibility Complex or MHC. So, let's see, what is it? Well, when we say Major Histocompatibility Complex, here comes three terms: major, histocompatibility, and complex. Here the second term is derived from further two terms. Histo, that is a Greek word meaning tissues. Our body's tissues, of course. And the other word is compatibility, that is a simple English word which means coexistence without any problem or conflict. And this term is applied to the MHC to describe their role in graft rejection or in tissues transplantation, as it has the ability to distinguish between self and non-self cells of the body. Also, this MHC was first identified by their potent or major effect on the transplanted tissues by the immune system, hence termed major. And lastly, it is a complex of genes. That's why it got the term complex in its name. So, after getting its brief idea from the term Major Histocompatibility Complex, now let's define it properly. Well, Major Histocompatibility Complex is a complex or group of genes that encode specific molecules which are responsible for the presentation of antigens to the immune system. So, simply what we learned from this definition? MHC is a group of genes that encode specific molecules, and those molecules are named as MHC molecules. And then these molecules are responsible for the antigen recognition, and then its presentation to the immune system. Here mostly it is then presented to T cells. So, this was the whole idea. This MHC has been given different names in different organisms. Like, in humans, MHC is also known as Human Leukocyte Antigen or HLA, and is present on chromosome number 6. So, here we can say, these two terms are interchangeable for humans. MHC genes or HLA genes that encodes MHC molecules or HLA molecules. Now you know these are same, so I will use any of these terms here in this video. If we look at this HLA or MHC region on chromosome number 6, we'll find that there are more than 224 loci for MHC genes. You know what is locus, right? The location or position of genes or chromosomes are termed locus, plural loci. These loci of MHC are classified into three major classes: MHC Class I, MHC Class II, and MHC Class III. The protein products of Class I and Class II play a vital role in immune system. And we know they are mainly involved in antigen presentation, where Class I MHC genes encode molecules that present antigen to CD8 plus T cells, and Class II MHC genes encodes molecules that present antigen to CD4 plus T cells. On the other hand, Class III MHC genes encodes complement proteins and cytokine molecules, but not directly involved in antigen presentation. Also, the main action of MHC Class III is still unknown, and thus, it is not very popular. We have a complete detailed video for you on MHC and antigen presentation. You can watch that video for detailed conceptual understanding. But, here in this scanimation, we are going to talk about MHC Class I only. So, what is MHC Class I? Well, MHC Class I is one of the three major divisions of MHC gene loci. And in this locus, the three main gene segments are HLA-A, HLA-B, and HLA-C. By following transcription and translation, ultimately these gene products produce MHC Class I molecules. Now, if we look at the structure of MHC Class I molecules, we get to know that Class I molecules are non-covalently associated heterodimers. That means it is made of two polypeptide chains that differs in composition. Here, these polypeptide chains are the alpha chain, and the associated molecule is beta-2 microglobulin. Here the alpha chain has further three domains that are alpha 1, alpha 2, and alpha 3. And the beta-2 microglobulin is then linked non-covalently to alpha 3 domain. Here, the alpha chain is considered a heavy chain, with a molecular weight of approximately 35 to 45 kilodalton. And this heavy chain is actually encoded by MHC genes, found on chromosome number 6. But the beta-2 microglobulin is a light chain, with a molecular weight of approximately 12 kilodalton, and is encoded by entirely different genes found on chromosome number 15 in humans. This beta-2 microglobulin is essential for the peptide binding function of MHC-1 molecules. The alpha 1 and alpha 2 domains of MHC-1 molecules spaced apart to make a special site called binding groove. And as name shows, at this binding groove, the foreign antigens or peptides gets attached. But this attachment depends on the compatibility between the amino acid residues at the binding site with that of the peptide antigens. So, we can say the amino acid residues that make up the binding groove are the site of polymorphism. That enables the MHC molecules to bind the plenty of antigens. For example, different HLA alleles produce different amino acids in the antigen binding groove, therefore they can bind a different range of peptide antigens. And so is true for HLA-B and HLA-C proteins. The peptide antigen has been said to fit into the groove like a hot dog in a bun. Other than this antigen binding groove, the opposite end of MHC-1 molecule, where alpha 3 domains pair up with beta-2 microglobulin, makes a base of MHC-1 molecule, from where they are anchored to the surface of almost all nucleated cells. That means MHC Class I molecules are found on almost all nucleated cells of the body. Like all virus-infected or tumor cells, leukocytes, and platelets. But are not found on non-nucleated RBCs. These MHC molecules, by passing their developmental stages through endoplasmic reticulum and Golgi apparatus, once get attached to the surface of infected cells. Together with endogenous antigen, that is the antigen processed inside the cells. And thus makes an antigen MHC complex. And ultimately present it to the T cells. And in the case of MHC-1 molecules, the T cells involved are the cytotoxic T cells or T cells with CD8+ receptors. Let's summarize what we learned till now. We've learned that MHC-1 are the specific loci of MHC genes. That encodes MHC-1 molecules. These molecules are heterodimers with alpha chain and associated beta chain. They are found on the surface of almost all nucleated cells. And over the surface, these molecules mainly represent endogenous antigen. And this presentation is for the cytotoxic T lymphocytes or CD8+ T cells. We have explained the definition and structure in a bit detail. Now we will discuss this last part, that is actually the role of MHC Class I. And we know this role or function of MHC Class I is called antigen presentation. But presentation to whom? Well, we just learned that it is presentation of antigen by MHC-1 molecule to CD8+ or cytotoxic T cells. I am repeating it again and again so that it gets fixed in your mind. This mechanism or antigen presentation is quite systematic and is done by a proper pathway that is named Class I pathway, as MHC Class I molecules are involved. Or it is also named as endogenous pathway. And why is named so? Because this class of MHC, that is MHC1, is known to present endogenous antigen to T cells. And what are these endogenous antigens? Well, endogenous antigen are those immune-triggering molecules that exist inside body's own cells. And these endogenous antigens are processed inside the cell and are presented by the MHC Class I molecules to the cytotoxic T cell via endogenous or Class I pathway, which we are just going to learn here. Class I pathway is adopted by MHC Class I molecules for endogenous antigens. And this pathway is essential for immune surveillance and defense against infected or abnormal cells. For our convenience, I am going to break this pathway into the small steps for better understanding. So, here the first step will be antigen processing. The endogenous antigen that could be intracellular pathogens, such as viruses, intracellular bacteria, parasites, etc. These pathogens infect the host cells, and by using host machinery, particularly in the case of viruses, they will start synthesizing their proteins inside the cell, along with the self-proteins. Now, there is a need to distinguish which proteins will be degraded and which will not. So, here comes the role of a protein, named ubiquitin. This protein is found in almost all eukaryotic cells, including human cells. And this ubiquitin is involved in tagging proteins that need to be degraded, and here these are the viral proteins. And thus it contributes to the generation of peptides that will later bind on MHC-1 molecules. Once marked with ubiquitin, these proteins are then broken down or degraded into smaller fragments by the specialized protein complexes, named proteasomes. This proteasome, along with many proteolytic enzymes, may cleave some of the viral protein molecules through various polypeptide and peptide intermediate steps, until peptides of 8 to 11 residues are formed. Now, after this antigen processing, next step is peptide transport and loading. The generated peptides of required length are then transported to the endoplasmic reticulum (ER) by the transporter associated with antigen processing proteins, simply called TAP proteins. TAP proteins facilitate the transport of peptides from the cytoplasm into the ER, which is the site for synthesis of MHC Class I molecules. After this transportation, next step is assembly of MHC-1 complex. Inside the ER, the MHC Class I heavy chain, that is the alpha chain encoded by genes within the MHC Class I locus, example, HLA-A, HLA-B, or HLA-C, is produced and folded. Then the heavy chain forms a complex with a small, non-covalently associated molecule called beta-2 microglobulin. The MHC Class I heavy chain and beta-2 microglobulin association stabilizes the MHC Class I molecule and allows it to be transported to the cell surface. So, now within ER, this newly synthesized MHC Class I molecule undergoes a process called peptide loading and editing. The peptides that were generated by proteasomes and transported by TAP to ER. are trimmed and further processed by enzymes within the ER to generate optimal peptide lengths for MHC Class I binding. And then the MHC Class I molecule binds to a specific subset of peptides that have high affinity for its peptide binding groove. After this peptide loading, next there occurs the transport to the cell surface. For this transportation, the MHC-I molecule, with the bound peptide, is then transported from the endoplasmic reticulum to the cell surface via the Golgi apparatus. As this Golgi apparatus encloses the MHC with bound antigen within a vesicle called endocytic vesicle. This vesicle then makes its path to the surface membrane and gets fused with it, exposing the peptide on the outer surface of the cell. Now, the last step that is triggered by the surface adhesion of MHC antigen complex is CTL recognition and activation. So now, when MHC-I molecule with bound antigen is presented on the surface of infected cells, the cytotoxic T lymphocytes or CTLs recognize that bond through specific TCRs that are found on their surface. So, when a cytotoxic T lymphocyte or CTL encounters a cell displaying a foreign peptide on MHC Class I, the TCR binds to the peptide MHC complex. This interaction between T cell and the MHC antigen complex triggers a series of signaling events within the cytotoxic T cells, leading to its activation and the initiation of an immune response against the infected or abnormal cell. So, all in all, by presenting pathogen-derived peptides on MHC Class I molecules, the Class I pathway enables the immune system to identify and eliminate infected or abnormal cells, thereby contributing to immune defense against intracellular or endogenous pathogens. So, I am hopeful that now you can easily answer what is Major Histocompatibility Complex or MHC Class I. And we are here to answer more such interesting questions, along with astonishing visuals to clear your immunology concepts. So keep watching this immunology series, and keep visiting sqadia.com.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.